Recording now. Yeah, perfect. Yeah, so I would like to welcome everybody to Computational Genetics discussion group. And today we're going to have Andries talking about quite important topic these days and in the last few years. And just for introduction, how important infectious disease prevalence is in animal breeding. So in last few years, we had the African swine fever virus going on around. And for example, in China, basically it decimated quite a big population. And I think something slightly less than 1% Chinese GDP was the total cost of that. So it's quite important topic. So with this, Andries, floor is yours. Go ahead.
Yes, thank you, and thanks for giving me the opportunity to present in this meeting. So my name is Andries Hulst. I'm a third-year PhD student now at Wageningen University in the groups of animal breeding and veterinary epidemiology. And today we'll be talking about my work on pathogen co-evolution following genetic selection for lower infectious disease prevalence. Yeah, I'm quite sure you're now quite aware of what an infectious disease is, and that there are multiple ways to combat infectious diseases. Of course, vaccination is very prominent now, but also social distancing was, well, now at least in the Netherlands, it's not. We got rid of it, but I think 2 or 3 weeks ago. But of course, the past 1.5 years, we all know that worked quite well against certain infectious diseases, but also antibiotics is an example of an intervention against an infectious disease. But of course, this is a genetics discussion group, and I'm working partly in animal breeding. So I'm mainly interested in animal breeding. And this year we published, together with Peter Bijma and Marty Jong, published 2 papers on genetic selection as an intervention against infectious diseases. And in those papers we showed that actually genetic selection is way more promising to combat infectious diseases or to reduce the prevalence of infectious diseases than was always believed. I will give a little bit of very briefly results of that. So this is one of the important graph from the first paper. So what to show here is that here there are 6 different lines, all for a different herd. And in those herds, we selected animals on low susceptibility. And so what you see is that the disease dies out after some time. And that was quite an interesting result, because the classical quantitative genetic theory suggests that you can never get rid of a binary trait. But apparently that is different for infectious diseases. So we also investigated why it's different. You can see that here. So it turned out that it's not only with selection that you can achieve that, but also with sorting. So on the left, you see, so in both figures here, the population consists of the same animals. The only difference is how they are put into a herd. So on the left, it's at random. And on the figure on the right, I first sort them based on their susceptibility, then put them into herds. And then what you see is that in these lines represent the 10% herds with highest and lowest susceptibility. So what you see is that in the lowest 10 percentile, the prevalence decreases to 0 when we sort the animals, and when the herds are at random, that doesn't happen. So what that shows is that it matters a lot who your herd mates are. So if you are in a herd with animals with low susceptibility, then it's probably likely that there is no infectious disease. So this is very comparable to the herd immunity effect following vaccination. So if we can select animals against infectious diseases, if we could reduce the prevalence of an infectious disease using genetic selection, then also the question comes, well, as we see with antibiotics, for instance, pathogens can adapt themselves quite well to interventions. So can that also happen when we select animals under genetics? So that was the starting point of what I will be mainly talking about today. So what we ask ourselves is, when can pathogen mutates invade in a genetically resistant host population? It might seem a little bit difficult to think, but was it when I started with this as well for me, but well, it helped a lot to set a very clear starting points for this analysis. So I did a deterministic analysis of a transmission model. And the starting point for that is a local host population. So for instance, a herd of cattle or pigs, which is endemically infected with a certain infectious disease. And that disease is caused by a certain type of pathogen, and we call that pathogen the wild type pathogen. And then animals in that herd are selected for increased host resistance to reduce the prevalence of the disease against that wild type pathogen. To make it a bit easier, for now we assume that this host resistance is determined by a single recessive allele. So there are only two genotypes here, so either the resistant genotype or the non-resistant genotype. And then the interesting part comes, because then when we have started selecting for resistance, then also mutants of the pathogen emerge, and they can invade into the host population. So the mutant that can escape host resistance, so that means that it can still spread in resistant hosts. But it's also, that's a bit, well, by definition, it's less successful in non-resistant hosts. So the resistant resistance mutation, or the escape mutation, sorry, comes of the pathogen comes with a cost when it infects non-resistant hosts. So I'll give an overview of the transmission model. So this is the well, very initial starting situation where we have only non-resistant hosts, so there's no selection yet, and one type of pathogen, the wild type, and that RNW that's there, that's the basic reproduction ratio of that wild type pathogen, and it's above 1. Well, if it needs to be above 1, because otherwise it cannot be endemic. So those who don't know, the basic reproduction ratio, that's the number of secondary infection caused by a single infected in a fully susceptible population. So in order for a disease to spread, basic reproduction ratio needs to be above 1. If it's below 1, the disease will die out, certainly. So then we start selecting for increased resistance. So because it was a single allele, we get one resistant host type. They have the resistance genotype. And in these resistant hosts, the wild type has a reproduction ratio below 1. I'll show in one of the next slides what happens if it's not below 1. But what I just said, if the reproduction ratio is below 1, we make sure that the infection will die out after some time. So this is the first step. So we now have a model with 2 types of hosts, but still only 1 pathogen type. So there are no mutants of the pathogen yet. But with this model, what I show here in this figure is the prevalence of the wild type pathogen in the host population over the frequency of the resistance allele. So across the x-axis, you see that well, from left to right, the resistance increases. So that means that on the left, we have a fully non-resistant population, and on the right, we have a fully resistant population. And the three lines, they represent different levels of the reproduction ratio of the wild type in resistant hosts. So that was the second one I showed on the previous slide. So what you see is indeed that when it's above 1, the disease doesn't die out, so the prevalence is even in a fully resistant population, it stays above 0. And when it's below 1, the disease dies out at a certain level of host resistance. And what you also see, and that's quite important, is that the lower this reproduction ratio is, the earlier the point of dying out. So the lower level of resistance is needed to eradicate the disease from the population. Yeah, and these points of dying out, we'll call that Q max, so the maximum frequency of the host resistance allele at which the disease can spread. We got quite a nice expression for that. This is also obtained for vaccination, because you have quite a similar approach there. So I think we can go on to the next step. And now it's getting interesting, I think, because now from the previous situation with 1 pathogen type and 2 host types, the certain point I allow pathogens to develop mutants, so that we get another pathogen type. This is an escape mutant, and that, well, can infect both types of host. And we model let's say the transmissibility of the escape mutant also through the reproduction ratios. So the reproduction ratio on the bottom of the slide of the escape mutant in resistant hosts, well, that is by definition, because that escape mutant by definition can escape the host resistance, so that is by definition it's higher than the reproduction ratio of the wild type, which means that the mutant can spread better in resistant hosts than the wild type. And also because we assume that there are certain costs to this escape mutation, as the reproduction ratio in, look at the laser pointer. So this one, the reproduction ratio of the escape mutant in non-resistant hosts is lower than that of the wild type. So that means that if you would have, let's say, a fully non-resistant population, that the escape mutant has a bit more, well, it will probably never invade because it's more difficult for it to spread in that population. So with this model, I could start to try to find out when these escape mutants can spread. So we'll take you through this figure. So this is what we had before, the prevalence of the wild type over the frequency of the host resistance allele. Now it will only show it for one reproduction ratio now, because otherwise it will get very complex, this figure. So one limit here for the arisal of the invasion of escape mutants is, of course, the part at which the wild type is not present, so above the Q max, so above the level of the frequency of host resistance for which the wild type can be present. So there the mutant cannot arise simply because there's no wild type, so there are no pathogens in the host population. So that's one limit. And the other one is a bit more difficult. So that's well, you can imagine that if this escape mutant, what I said, it spreads better in resistant than in non-resistant hosts. but it also competes with the wild type because that's already present. Because it spreads better in resistant hosts, it probably needs some degree of host resistance before it can invade. And to derive that point from which it can invade, so we need a different reproduction ratio. So that's not a basic reproduction ratio, because the basic reproduction ratio is defined for a fully susceptible population. But I will call this the invasion reproduction ratio. So that's the reproduction ratio of the escape mutant in a population that is endemically infected with the wild type. So that's the population that we have, in fact, because that's endemically infected with the wild type. So what you see here in this formulation, you see the two reproduction ratios of the two basic reproduction ratios of the escape mutant in non-resistant and resistant hosts. And they are multiplied with the fraction of susceptible hosts. So that makes sense, because when you have a fully susceptible population, as is the case for the basic reproduction ratio, then this fraction is of course 1. But now we don't have a fully susceptible population because a part of the animals is infected with the wild type already. And then we need to multiply these reproduction ratios with the fraction of hosts that is still susceptible. And then at the level of their resistance status. So 1 minus Q squared is the total fraction of non-resistant hosts, and the P and W is the prevalence. So the fraction that is infected with the wild type already. And similar here on the right for the resistant hosts. So this R invasion, that is the dashed line in this figure, and the scales on the right y-axis. So I put a dotted line at 1, because also for this reproduction ratio, it holds that it needs to go above 1 for the mutant to be able to invade. So what you can see now is that we have, well, here on the left, a part in which the reproduction, so the invasion reproduction ratio is below 1. So that means that the mutant, well, it can arise because there are wild type pathogens, but when it arises, it cannot invade because it's outcompeted by the wild type. But there is also an area in between these two limits. And that's what I will call the invasion window. So that's the area, or the range in host resistance, in the frequency of the host resistance allele, in which the mutant can arise because there are still wild type pathogens present, but it can also invade. So that means that the host resistance is high enough for that escape mutant to be able to infect enough hosts so that it can spread. And that's of course the area of interest, because at that point there the problem is that when you are selecting, and you will probably increase the frequency of host resistance, so in this case 2.7 or so. So when you stay there, you see that the prevalence has gone down quite a bit, but then at a certain point a mutant might arise that can still infect other animals, and then your whole effort put in the genetic selection is well, let's say, diminished by the escape mutant. Yeah, so this is a little bit back to what we saw before in one of the first figures with the lower reproduction ratio of the wild type in resistant hosts. So in fact, this lower reproduction ratio, that means that the resistance is more effective. So that can either be that the genes are well, the resistance genes are stronger, or what also can be is that genetic selection is combined with other interventions that reduce the transmissibility of the wild type. And what you see then is that it's not only that there is less resistance required to eradicate the pathogen, but it's also the invasion window is smaller in that case. So that means that there is a smaller range in which there is a risk of escape mutant invasion. So far so good. I hope now I'm going to make it a little bit more difficult even, because so far I assumed that hosts could only be infected with one pathogen at a time. So either with the escape mutant or with the wild type. But often what we see, especially often what you see is that hosts can get infected with multiple types of pathogen at a time, or even But that is because these of course here we have a mutant of the wild type, so that means that there is probably already some immunity in the host. So once a host, what will happen then is that an already infected host, for instance, with the wild type, that's the line above, that gets across an escape mutant, and that escape mutant can probably also infect that host, and then that host is infected with both types. But of course, these pathogens are related because the mutant is, the escape mutant is a mutant of the wild type. So that means that probably the host already has some immunity against this, or resistance against this other pathogen type as well, because it was infected with a related pathogen type before. So I call that cross resistance. We included that into the model. And the effect on the invasion window, so that's in this figure. So the smaller invasion window, that's for an R, that's the cross resistance of 1. So that's full cross resistance. That's the same as we had before. So in that case, hosts can only get infected with one type of pathogen. But you see that with decreasing cross resistance, the invasion window gets wider. And here, even when there is no cross resistance at all, it covers the whole range from 0 to the point of dying out of the wild type. And that's mainly because of course, set some values for the reproduction ratios. And the escape mutant has a reproduction ratio of above 1 in non-resistant hosts. But what you clearly see here is that although it is above 1, if there is cross resistance, that doesn't mean that the escape mutant can always infect these hosts, because it is outcompeted by the wild type. So only if there's no cross resistance, so that means that, it doesn't matter it doesn't matter at all whether an animal is already infected or not with another type. At that point, we see that the mutant can then infect all susceptible hosts, all susceptible hosts to the mutant, so also the ones infected with the wild type. So this cross resistance is important to account for, because if you don't do well, it's probably that you underestimate the width of the invasion window. That's what we see here. There is a difficulty, and that's that. If we have an endemic disease, we probably can estimate quite well the reproduction ratios of this wild type, because that's already present for quite a long time. We probably can also estimate the effects of selection on that wild type, and what the reproduction ratio in the resistant host is. But most likely we cannot. When you start selecting, you of course never know what escape mutants will arise. First, in this figure, I still will show again the effect of the reproduction ratio of the wild type in resistant hosts. So in fact, the strongness or the effectiveness of the resistance gene on the width of the invasion window. So now for a full range of reproduction ratios. So you can see that on the y-axis is the increasing reproduction ratio of the wild type in resistant hosts, and you can see that the invasion window increases quite a bit with the increasing reproduction ratio. And that makes sense because the better the wild type is well, what you can also see here, that's the part on the right. So this line here, that's the point of dying out of the wild type. So what we see is that the invasion window is mainly wider because it's more difficult to get rid of the wild type. And now the other part I was talking about, and that's about the effect of the different reproduction ratios of the escape mutant. So when you start selecting, you of course never know what mutants will arise. You can do some predictions as what I did, that it will probably be less, well, the reproduction ratio in non-resistant hosts will probably be lower than that of the wild type. And of course, because it's an escape mutant, it will have a high reproduction ratio in resistant hosts than the wild type. But how much higher it is, well, in fact, you don't know when starting selection. But that makes it difficult to construct these invasion windows. So in this figure, I show the invasion window as a function of the reproduction ratio of the escape mutant in non-resistant hosts. So again, the greener line on the right is the frequency of host resistance at which the wild type dies out. So that's the upper bound of the invasion window. And the dotted or the dashed lines on the left, that are the points as with the R invasion, which we saw before, becomes above 1. So that's the lower bound of the invasion window. And then for the 3 different levels of cross resistance. So what you see here, that when the escape mutant is, well, let's say when the costs of resistance are very high, so that means that the escape mutant is not well able to infect non-resistant hosts, and we have a very narrow invasion window. So a lot of, well, a very high frequency of host resistance is needed for the mutant to be able to invade, but that high frequency is also already almost the point where the wild type will die out. So then it cannot arise anymore. And what you see is then with increasing reproduction ratio, the window becomes wider and wider, and especially when it gets above 1, it can get very wide, almost covering the whole range of the whole frequency of host resistance. And that's also what we saw before, because this when the escape mutant is also able to infect non-resistant hosts, and especially when there's not much cross resistance, then it can easily invade by infecting these non-resistant hosts already at when the frequency of the host resistance is low. So when there are a lot of non-resistant hosts. So the next figure is then, of course, for the reproduction ratio of the escape mutant in resistant hosts. So that, yeah, well, that's in fact, well, let's say how well the escape mutation works. So the higher the reproduction ratio, the better the mutant is able to escape host resistance. And here it's again the same. We have on the right the point where the wild type dies out, and on the left the dashed lines that are the lines from which the escape mutant can start to invade. So when we started at the top, that's the one where there's full cross resistance. you see that this invasion window that becomes wider and wider with increasing reproduction ratio of the escape mutant in resistant hosts. And it's the same for the when the cross resistance is lower, but now it becomes even more wide or wider. And an interesting thing here is, of course, this shape of the lower line. Because there it seems that in fact, yeah, well, the invasion window is well, at least it covers the whole range of frequency of host resistance, and that's because what we saw before as well. I assumed here that the reproduction ratio of the escape mutant in non-resistant hosts is above 1. So that means that when there's no cross resistance, that it can spread always. But then what you see here is when the reproduction ratio of this escape mutant in resistant hosts is below 1, in fact, the selection also works against this escape mutant. But of course, till a certain point, and you will still have problems. Yeah, well, the question is then what the relevance of this is, I think. But the point is that well, when the reproduction ratio of the escape mutant in resistant hosts, well, there might be a situation when that is higher than that of the wild type. So that's maybe, well, not exactly what's shown here, but then you might get a point at which you can also eradicate the escape mutant. But it's maybe not very relevant. Some discussion on these results. Well, yeah, what I said, the reproduction ratios of the escape mutant are unknown, so that makes it difficult to predict. And also, of course, the cross resistance is unknown when you start. So that makes it difficult to predict what exactly the width of the invasion window will be. So in what area or in what frequency of host resistance you will be at risk. So that makes it important to, I would say, to get as fast as possible to the eradication point, because from that you're sure that there are no possibilities to invade. Another point is, of course, that disease resistance is, well, I think quite often polygenic, and here we assumed a very simple genetic model. What's already hypothesized on that is that when disease resistance is polygenic, it might also be that the pathogen will need multiple mutations. And that would probably make it more difficult for pathogens to adapt to genetically selected hosts. And that would, of course, be nice, because that means that it's more difficult for pathogens to invade, or for mutants to develop mutants that can infect resistant hosts. And probably the most important question, what to do with these results. Well, what I just said is that when you start, you don't know what mutants will arise. But the good news is, I think, especially in livestock, that it seems quite reasonable to get to, or feasible to get to local eradication. If you wanted to do that through genetic selection, then it's also very important to take the herd composition into consideration. So the frequency of the host resistance allele, what I showed in the figures, you can of course increase that by increasing the frequency of resistant hosts. And if the fraction of resistant hosts is high enough, then we can probably eradicate the disease and prevent mutation or invasion of escape mutants. So it's probably not very feasible to achieve that through the gradual selection that's usually applied, but you think of sorting animals. but especially for pigs and poultry, it also often the herds are replaced at once. So then, especially then, it should be quite feasible to get a herd in which the fraction of resistant hosts is high enough. And when that's more difficult, you can also think of creating subherds, for instance, so that you have one herd in which the resistance is high enough to eradicate, so that there will be no infection at all. And in the other herd, the resistance is that low that you are sort of on the safe side of the invasion window. But then, of course, it's very important that these herds are strictly separated and that there's no risk of spreading pathogens from one to the other, because then you still might run into problems. Yeah, but when you do it, let's say in a multi-trait approach, that might not work, probably, because then the improvement of resistance will probably be so slow that a herd might spend a very long time in the invasion window. So then you still might see that the prevalence goes down, but then probably at a certain point, pathogens might evolve mutants that can escape the host resistance. And then, well, let's say your selection is not effective anymore. And also a thing to account or take into consideration is that when increasing resistance gradually, also when it goes quite fast, when the herd was endemically infected before, it might take some time for the pathogen before it has faded away, although the resistance is high enough to eradicate. And then, of course, as long as it's present and it infects animals, it might still be that mutants arise. So then it would be important to take out infected animals, for instance, to prevent that you still get escape mutants, although the wild type is suspected to die out. Yeah, so the conclusions, well, what we mainly see, I think, in from these results is that to prevent the invasion of escape mutants, it's important to increase host resistance quite rapidly, or probably jump over the invasion window at once by sorting animals or so. And another thing is that although we can maybe not influence the strength of the resistance allele, we can still influence the width of the invasion window by making sure that it's very hard for the pathogen, for the wild type to spread. So when you combine genetic selection with other interventions, for instance, that might decrease the reproduction ratio of the pathogen, makes it easier to come to eradication, and also make the invasion window of possible escape mutants narrower. So that was my last point. Combined breeding with other infection management can help a lot in this, I would say. So that was it for me. Thank you for your attention. I'm very curious for any questions or suggestions.
Thank you very much. Great talk. So we already have one question from Andrea Wilson. She's here. Should I read it, or Andrea, would you like to do it yourself?
Yeah, I can ask myself. Thanks for the nice presentation, Dries. My first question was just to clarify again. So you assume that resistance is full resistant to becoming infected, right? In your study, is that correct?
No. No, so the only thing we assume is that well, there are only two host types, so animals are either non-resistant or resistant. But when they are resistant, that doesn't mean that that they cannot get infected at all. So that's what.
Okay.
Very back. Yeah. So what we see in this figure, for instance, so it only means that the reproduction ratio in those resistant animals is lower or below 1, but they still can get infected.
Okay, because there's formulas, right? Which is a bit like the effect size of the resistance, and then you can calculate the reproductive ratio based on the effect size and then the proportion of individuals that are resistant from the epidemiological literature. So that's why I was a bit confused whether whether you use that or how you stipulate, because you said you don't know the reproductive ratio, but you actually do, because if you know the effect size of the allele and you know the proportion of individuals, you can actually calculate it.
Yeah. I mean you don't know the reproduction ratio of the mutant, so of the escape mutant. So of course when you know what your resistance allele, how strong that is, you can indeed know how effective it is against the wild type. But the mutants, they are of course not there. So
okay. So you just basically this is then the trade-off between, because there's in the literature they often refer to the trade-offs between. So basically in the population that is resistant to the wild type, they are more susceptible to the mutant. I'm just trying to match it what I know. So this is very interesting. But because my question is kind of what you've shown, there's a lot in the vaccine literature, right? And I'm sure you're quite aware of that.
Yeah.
And what is different from what you derive to what they know in the vaccine, so the models in the vaccine literature? Because that your Q, it could be the vaccine coverage, so the proportion of the resistance alleles is kind of the vaccine coverage. That's why I'm trying to make sense of what exactly is new there, what is different, how, when we talk about genetics, can we just adopt what they have in the vaccine literature, or do we need to start new? And I'm just trying to get my head around this, because you approach it a bit different. So sorry for my confusion, but maybe it's clearer to you. Because there's loads of vaccine studies to say, what does it take, to prevent an invasion? And especially now with COVID, it's so relevant. How many individuals do you need to vaccinate so that your new strain can't invade? So there's loads around at the moment. And with breeding, there's nothing around. So how is that different?
I see your point. So indeed, with this, let's say this very simple genetic model, it is of course, well, you still have, when you vaccinate, you also have 2 host types. So indeed here, it also just 2 host types. So that's very comparable. So I think from this point, it's mainly the new thing is that we got into it from a different angle. So from the breeding side. Which
is always good. So I think it is good. But I think it's worse when I don't know if you've published that already, but when when you think of publishing it, just, go a little bit in the vaccine literature, because the mindset is very similar. What's new, what's different, what's the new angle?
Oh yeah, sure. Yeah. I see the point. I see that Mart wants to add something maybe.
Yeah, Mart, please go ahead.
Yeah, if I can, do I have my video? Oh yeah, I have my video on. I don't know whether I be seeing visible, but okay.
Please go ahead.
Yeah, so the point is that much of the vaccine literature is based on humans, and so this whole idea to be able to bring the reproduction ratio below 1 is often not addressed. So we looked at one of these vaccine papers, and so they really only look at escape mutants or resistant hosts, but still there's the reproduction ratio of above 1. and then they look at different aspects of the vaccination that would be selected against, but in animal populations, especially in farm animal populations, we can bring the reproduction ratio below 1 in certain herds, and that is, I think, the main different thing here, because then you have a really different kind of discussion where you try to eradicate a pathogen from a local population. Whereas in human vaccines, you often it's not thought about of eradicating the pathogen by vaccination. I think that's the main, that would be a big difference, I think.
I was
Do you
wondering
think,
also,
Andrea?
I was wondering if it's also because here you bring in selection as well, right? So that the time scale is also a bit different between selection and vaccination. That may be worth comparing, because your invasion window, that affects your invasion window. Maybe that's another aspect to look at.
Yeah.
Because,
Yeah, so I see your point, and we're wondering about this. So we should really look at it. But what I've seen from vaccine is not that they really consider the possibility that you really can eradicate an infection. That's because they look at a very large mixing population. And here you have local populations that only mix locally, and you can do something locally to the local population and eradicate it locally.
Okay.
And there I don't know some literature about escape mutants in animals. Yeah, but yeah, so maybe you know, but could point us to. It's an interesting discussion. Yeah, so there's certainly a parallel with vaccination or other interventions here. Yeah. But yeah, indeed, also the time scale is a difference. Yes.
Thanks. Good. Yeah. I can suggest a few papers because I looked a little bit into that too, and
Okay.
so I haven't got my head around it. So that's why it's very interesting for me to ask you.
Yeah. So you're not sure that. So one of the papers that we looked at, I'm so bad at names, was Parker. Was one of the.
It was Parker Majori, I think.
Okay. Yeah. And so there they explicitly considered only the situation where the R would still be above 1 after vaccination. And so they look at small fractions of the population vaccinated. So they have much less of the indirect effect of vaccination, because they only have a low percentage vaccinated. Like in influenza, you only vaccinate people at risk. And so it's a very small proportion of the population. So you don't really have an impact on the infection dynamics there. But maybe now with SARS-CoV-2, there will be more models where they also consider the effect on the infection dynamics, because there seems to be a clear effect on that. But I'm not aware of it yet. So,
I just know Rustam Antia's models and, Martin Novak. So they had looked a lot into the evolutionary aspects. So there's a lot of theoretical literature around, but it's quite hard to get your head around it.
Yeah. So that's more the evolution. I know. Yeah. But so that's more evolutionary without population dynamics. And this is population dynamics and very simple evolution. Combined, So this is, I don't know, but certainly we should have a careful look at that, and they're quite clever. So I mean, but they have so yeah. Okay, that's of course that's something to think about. Yeah. Nice.
Okay. So we have a good discussion opened. Go ahead. Any more questions? While people think about the questions, can I maybe ask from kind of breeding perspective, can you actually recognize some kind of more resistant genotypes in your population? And in that way that you can actually breed for resistance, if that's even possible, or if you can have some kind of indicator or proxy trait for resistance.
Ooh. I'm not very sure about that, but I think, yeah, there is of course still already something known about that, but often not, probably not what reproduction ratios are. But I think, yeah, when you want to know, you should do some experiments, I would say, or find out in the field. That should also be possible. But then there's of course always a risk that these mutants may arise.
Okay. We have another comment in the chat. Hi Mark, the flu vaccination of human depends on the country, in contrast to the Netherlands. Here in UK and USA, they vaccinate many people, including all children in primary and secondary schools. Great for modeling. Then we have answer. I know about the idea to vaccinate children, but is it really done? In Netherlands, getting hospital staff vaccinated against influenza was difficult. So yeah, these are the comments. So okay, any more comments? Please go ahead. We have 7 more minutes. Okay, another comment from Andrea. In livestock, host-resistant relates mostly to increased resistance to develop disease rather than the resistance to becoming infected. Do you think your model results are also relevant for this wider context of resistance? And you can either read again from the chat, and Andrea is also here if you need more clarification on the question.
No, that's an interesting point, I think, because that's indeed what's mostly called resilience. I think that animals probably still can get infected, but not they don't develop disease. But then I think there's always a risk that well, it depends a bit on how this pathogen works and how the transmission works. But often with these things is that it might also allow pathogens to get more and more virulent. So, for instance, if the animals don't develop any symptoms, then the pathogen can also use that to become more and more violent, because also pathogens might need symptoms, for instance, coughing or whatever, to transmit themselves. And in the end, I think the pathogen with the highest fitness is the one that infects most other animals. So I think when just breeding against, let's say, clinical symptoms, I think that would be a risk in terms of mutations of pathogens, I would say. And of course, then there is still transmission around, so then you can probably never, well, you can never get to eradication, for instance, then. So that's
yeah, I was thinking along the same line. Thanks for that, because I just thought, you know, if often resistance in animal breeding is really meant that they are resistant to, to the disease, to developing disease or die. So imagine that a more resistant host is less prone to die, so it will probably then so how would that affect the reproductive ratio? It could actually increase it. So I'm just wondering how that affects then the invasion window. It could actually backfire, right? So that the resistance, it's the opposite effect almost. If resistance has the context of prolonging the lifetime of an animal that would otherwise die now can survive longer, and whether it's just the inverse then.
Yeah, indeed, that's indeed. I think it can go. Yeah, it depends a lot on how this pathogen, how the transmission works. I think. Yeah. I think if animals die, then of course there is, or when they normally die, then indeed when they don't die anymore after you select, and you only increase the transmissibility of the pathogen. I think. So that's indeed a risk then.
Yeah, I know much more about vaccination than about breeding, so this is interesting. But for vaccination, it's clear that if you only protect against clinical disease, then you have more probability of escape mutants. So you have bacterial diseases, for instance, where children are. Whooping cough is an example. So yeah, whooping cough. So I don't need to look in my head for the scientific name. But yeah, so it's clear that this vaccine, there's vaccine escape against that. So you vaccinate for a long time, and it only protects against clinical symptoms. So the bacterium still transmits, and then at some point you have a more virulent strain affecting children. So it was a couple of years ago in the Netherlands we had outbreak of that, and so then they changed the vaccine. Yeah.
Okay, thank you for the discussion. Do we have any final comments? We are approaching 5 o'clock. So, okay. Seems we have no more questions. So I would once again like to thank you for being part of this computational genetics discussion group. It was a great talk, something slightly different from our usual talks on genetic genomics and breeding. So thank you for that. And as you see, it seems very nice discussion that your talk sprung. Yeah.
Yes, thank you.
Thanks to everybody. Yeah. And see you in 2 weeks' time, I think. Yeah.
Thank you very much.
Thank you.
Thank you.